as he said, I'm going to be talking a bit about this 3D spatial transcriptomics, which we are working on in here at SDU in Odense. So first up, the CellX node here in Odense is kind of, or the CellX management group members are Susanne Mandrup and Jonathan Brewer and their groups here. And we are kind of working on making these different systems for a technique called Murphish, which I will talk a bit about later. So we'll set this technique up and allow for spatial transcriptomics in different scenarios. So there's this super resolution spinning disk, which we will hopefully get within the next few weeks. That will allows us to scan large tissue slices and do it well in 3D. Then we have some localization microscopy through storm microscopy, and this will allow us to get a better resolution, but this will also limit us in the field of view, so we will have smaller areas in, for example, tissue slices or cell cultures. And then finally, we have this new microscope called MinFlux that allows us to get down to the 1 to 2 nanometer scale and really do some subcellular resolution on individual cells. So we're looking forward to actually applying this technique on these different systems here in the coming years. So if you didn't know by now, we are working with this imaging-based spatial transcriptomics, and basically we use or we take a series of images and we then can extract this single cell expression data from it. We can then pull the data out and do some more classical analysis, for example, cell typing, and then we can project this information back into the tissue context of the given sample. And to do this, there are many different techniques for doing this, but we have chosen to work with this method called Murphish. This is multiplexed ever robust fluorescence in situ hybridization. And just very briefly, the way it works is that, for example, we have this tissue slide here on a cover slip. We can zoom in on a single cell, and inside this cell, there's these different RNAs that we want to kind of target. So we look for some, or we want to know about the expression spatially of different RNA species in this sample. So if you zoom in on a single RNA strand here, we hybridize the RNAs of interest with this encoding probe through this target sequence. And this target sequence is flanked by these readout sequences that allows us to sequentially label this RNA, so we can get the exact location of it in this sample. And the way it's built up is through these hybridization rounds, which will give us a corresponding barcode. So for example, here in the first hybridization round, we flush in some fluorescent probes. They attach to this RNA or to the encoding probe on the RNA, and then it lights up inside the sample, and we can kind of pinpoint the location of that. So this position will be given like a 1 in this first position of a barcode because it's visible here. And then to remove the signal, we can photo bleach it, or we can chemically remove the fluorophore, and we can kind of carry on with the next round of labeling. And in this case, this particular RNA wasn't really labeled, so this corresponds to a 0 in the barcode. And also, for example, you can see here we targeted some other RNA species here, and then we can kind of just carry on with the experiment, do all of our labeling of the RNAs of interest, and we end up with this final barcode for this particular RNA spot here. And talking about the barcoding scheme here with this technique, it's very clever because beforehand we have designed this codebook consisting of different targets of interest, giving them each a barcode. So we know what we label in each round, which we can then afterwards look back into and then decode kind of this barcode that we get for the positions. So just to give you an example, this experimentally obtained barcode here, we can try to compare it to some of the barcodes that we designed beforehand for different genes. And we can see in this case it matches perfectly to this keratin 14, for example. And it has a further distance, we call that a Hamming distance, to, for example, another gene. So we can conclude here that this is keratin 14. But this technique is very error robust. That's also in the name of the technique. And this can be seen here, where we have a single bit error. This could be, for example, a mislabeling or something went wrong during the experiment. And now kind of have a unmatched position here, but we can still say that this experimentally obtained barcode up here is relating closest to keratin 14, so it must be that. So it's a very clever way of reducing the error in this sequential labeling. And to kind of give you another view on the data that we get out of this is kind of this image stack of the sequentially imaged rounds. And then we can kind of decode the RNA identity of all these detected spots in the different rounds, and then do some more classical analysis downstream afterwards. And to show you some of our current data, we kind of always get these images of the probes because that's the genes we are looking for. And also besides that, we are getting these fiducial beats in another channel. And these are used to kind of correct the drift that happens over a long set of hybridization rounds. So I hope this translates well onto Zoom, but you can kind of see this small drift here in between the rounds. You can also see different spots corresponding to different labelings during the rounds. And you were kind of promised 3D spatial transcriptomics. So here's an image of the kind of 3D data that we get. This is the current data that we get from a wide field microscopy technique, but in the future we hope to, or we will get a bit better data spatially with some of the new microscopes. So to kind of do this experiment, there's a lot of different things going on. And one of the things that we have developed is this automatic and mobile fluidic system that we can couple to an imaging system of our choice. So this is the image or the fluidic platform here, and then of course the microscope over here. And we can kind of program the fluidic system ourselves, so we can do whatever kind of experiment design that we want in this case. And we can also just lift it up and then go to another microscope if we want to look at another sample with another technique or at another resolution. So it's quite a flexible system that we have built. But as I said, there's a lot of different things with this method, and there's a lot of wet lab work, which I won't really go into detail about. Maybe some other time somebody else can talk about that. But I'm heavily involved in this analysis part of MERFISH, and as you saw before, kind of the raw data that we get are the images of the probes, and then the images of these beads, and then also some kind of nuclei staining. It could also be some kind of cell staining to allow for some segmentation afterwards. And then I have outlined some of the common steps to kind of get this actual spatial transcriptomics data. out. And kind of there's a big challenge in that the current tools available for this are not really standardized, and they are highly specific for the setup in the given lab that wrote the code. That also and also the documentation of these tools are not really that good, so it's incredibly hard to use these tools because of the lack of documentation. And also in our case, there's a lack of tools that can do complete 3D analysis. So to kind of solve this problem, we have written some of our own scripts in Python mainly. And then these scripts can then be combined to a final pipeline using, for example, SnakeMake. So I'll try to take you through some of these steps, these different analysis steps, until we get some actual spatial transcriptomic data. So at the beginning, we kind of want some pre-processing of our data. Our images can be very noisy, or there might be a high background from tissue. So we need to remove some of that. And also we would like to, almost always, we would like to do some form of deconvolution. So we get from these kind of blurry punchboard functions of our point light sources to some more precise spots that we can actually detect later on. So once the data is nice, we can kind of move on and, for example, do this drift correction I talked about. So we use these beats and we kind of track them all throughout these hybridization rounds in 3D. And then we can kind of correct the images afterwards. So you can see there's a bit of drift here. And then we can calculate some very basic statistics for it and then apply a correction to the drift that happens. That leads us onto one of the more important steps here. That is spot detection. And there are numerous different approaches, each with their own flaws and upsides as well. But for example, here we would like to detect all of the spots in the image, and we do this for all the rounds. This gives us this 4-dimensional spatio-temporal data of the coordinates over time for each point detected. And we would like to do some decoding of this to actually get the genes detected in the sample. So here I have developed a few algorithms taking some machine learning parts into it. So for example, I do some clustering through an algorithm called dbScan and cluster all the points found. And then based on this clustering, we can create some barcodes using the rounds of the clustered points where they were detected in. And then finally, we check these barcodes up against the design codebook, and then get out the gene that they correspond to. So just to give you an example, it might be a bit hard to see, but here there's a coloring in 3D of points clustered, and then we can kind of zoom in and then get a coloring based on the genes here. And then we can kind of compute the centroid of each cluster to get the precise location of each detected RNA in this case. So now we kind of have the spatial RNA data, but we would like to get it single cell level as well. So we need to do some kind of cell segmentation. And here I've previously done some work with this deep learning tool called CellPose, where we just take some nuclei staining that we have and then use the density of the detected readout probes that we have, because we expect that they will be most dense inside the cell. Of course, depending on which RNAs you detect, this can be better or worse, but it results in some quite fine cell segmentation in this case. So we combine this, and then putting it all together, we kind of get this detected RNAs in a single sample, then with this three-dimensional cell segmentation as well. And then afterwards, once we have this single cell RNA data with the spatial coordinates, we can then move on and do some other analysis. That might be a talk for another time, but we can also choose to kind of put it into a tool so we can actually visualize what is going on inside our samples and kind of really exploit this three-dimensional component of our data. So during my master's, I developed this tool for actually visualizing a sample with some different components here. I can just try to see if these videos work. You can kind of, it's an interactive tool, so you can kind of zoom in and hover over some of the detected RNAs and get their position. And it's also possible to select the single cells, and then kind of you get an update of a point of a plot of the different RNAs kind of detected in there. And depending whether you combine other analysis inside this program, you can get some more statistics out of it, or heat maps of the different areas selected. So yeah, that was pretty much it. There's a lot of people working on this MERFISH here at SDU, and I'd just like to thank them all. Just so you know that it's not only me that's working on this. I'm just a bioinformatician in this case. So yeah, thank you for listening.